All right, let's begin. So I'm like I said, you guys can take your notes online or if you want to take it on paper. I do suggest grabbing the booklet out of the box so that way you have a way to kind of write notes or you can write on the notes or see the notes as I'm going through them today. Does anybody need a biology booklet of notes? I remember I needed to print a few more because we had some people add it to our class. Does anyone need biology notes? Are we good? Does anybody need a variation checklist? We good? Presley, the phone. I'm happy to charge it. You can't use it. All right. Cool. My deck today, I just wanted you guys to have the opportunity to look through those project words. You did cover these guys last year, but I will refresh it as part of today's lesson, so don't worry about that. Today, we are going to be defining gene allele. Two really important words, and we have to be really careful about when we use each one because they will mark you down if you choose the incorrect one. The other thing we're going to be doing today is we're going to be talking about how mutations create new alleles. Cool. And then there was two things I need to remember. I checked, I think everybody for slide number request, so I think I'm done with that. The other thing that I have to ask you guys, I have misplaced the string that was used for the phenotype and I've also misplaced my mandala coloring book, which is one of those prizes. If anyone has seen them, can you please let me know because I don't know where they went. And I have spent my own money on a lot of these things that I use. So please let me know if you know where it is. Cool. All right. Shall we keep going? Okay. So the lesson plan for today, it looks quite heavy. It looks like there's quite a bit of stuff there, but actually a lot of these activities are a little bit short. It's also going to be a little bit heavy on the notes for the first half because I need to go through all those definitions. But to make it more interesting towards the end, what we're going to be looking at in the second half, I don't want to say second half, we're going to be looking at some diseases today. I'm going to show you guys various videos, kind of show you the impact of those diseases. And all those diseases tie into what we're talking about when it comes to mutations. So we'll see sickle cell anemia, we'll see cystic fibrosis, and we'll see Huntington's disease as well. These research aren't very long. It's just quick questions, just to kind of reinforce what we talked about with the mutations. I do have to consider the following question, and then we'll wrap up today with a nice little arts and crafts, where you guys can make yourself a little DNA bracelet, kind of like we did with the bracelet. All right. So success criteria, a lot of people are going to tick off today. We're doing a lot of work with vocabulary, so explain the difference between genes and alleles. When I see a typo, let me fix that. It's going to annoy me. Mask, please. We'll define the term mutation, and we'll look at the different examples of that. We'll see how mutation affects an individual, and how it can change the evolution of an organism, and also we'll look at genetic and somatic as mutations as well. So that's what we're doing for today's lesson. So to start off with, we need to think about the vocabulary that we're looking at. The first thing is going to be what a gene is. So when we're talking about a gene, we are talking about a section of DNA that's coding for that trait. So in this image example, you can see how this is a strand of DNA. We talked about it in our last lesson. And with that strand of DNA, we have a certain location, and that location is referred to as my gene. So that is the coding for my trait. So the various traits include things like eye color, hair color, height, nose shape. All of those things are examples of different genes we have. Are we okay with that so far? So gene as a definition is that section of DNA. Again, you don't need to worry about writing all this down because it is printed in the booklet and it is on my Google site as well. Cool. So in order to understand how the gene works, we need to see how it links from gene to protein to phenotype. So what we looked at in our last lesson is we learned that the protein and the phenotype are linked together. because the protein gives that particular trait. But now we need to understand how that gene tells the protein how to be made. So, what happens in our DNA? Remember how I said there are the 4 nucleotides, the A, T, G, and C? With that, the body reads the DNA in sets of 3. That set of 3 tells me what amino acid to grab. If you guys remember, we saw that big table with all the different amino acids. There are about 20 that the human body uses. And it's really important that those amino acids get into the right sequence so that protein can fold correctly. So we refer to this reading sequence as our codon. 3 nucleotides, and that tells me what amino acid I get. So it reads that 3 set first, it then moves on and reads the next 3, and then the next 3. And that's how we get our sequence. Really important for the protein to fold, and then we know that protein is going to result in that physical break. Are we okay with that so far? As I get? Good? All right. Now, that's all we say about this. If you are interested in taking an extra external and you want to do Bio 2.9, that's called gene expression. You learn how that actually works in a more detailed fashion. So just keep that in mind if you are interested in that kind of stuff. Cool. All right. So the next question is, I have one gene, I have a section of DNA, but how does it result in multiple traits? So, like I said, we have a section of DNA, and we refer to that as our DNA that says this is the trait. So, for example, we are dealing with a trait for eye color when I'm in that location of my DNA. Now, what happens with that sequence of DNA is that even though it's all the same section, the sequence can be different. So, when I say the sequence can be different, I mean the letters that are in that section could be different. So we see in this red color here that this second nucleotide has changed. We have one that's a G, a T, a C, and an A. That then results in the different eye colors. So the different eye colors are an example of the allele. So alleles are just a variation in that DNA sequence. You need to be really careful which word you choose when you're writing up your answer, because you need to show competency in how to use the language. So just watch out for that. Any questions? You guys should have hopefully come across both of these words last year. We're good? Okay. So the next thing we talk about then is how do these different alleles arise? So we know that DNA is a code of life, and in order for us to have that code of life, each cell needs to have that DNA, so that way it has the instructions. So in order for each cell to have that DNA, we need to copy it. We need to replicate it. Now, this replication occurs both in mitosis and meiosis. Did you guys do that last year? No? No? Okay, that's why we have it, because we're going to learn about those cell divisions later in this topic. Just know during those cell divisions, when you copy the cell, you need to copy the DNA as well. Now, what happens sometimes in that copying process is a mistake is made, and the nucleotide changes, and it's not the same anymore. That is what we call a mutation. You guys may have heard the word mutation come up before in various kind of things. This mutation is permanent. So the moment I make a mistake in that DNA sequence, all the future cells that come from that cell will have that mistake as well. So it gets passed down. These mutations can vary. Sometimes these mutations are beneficial and they're helpful to the organism. Sometimes they're harmful and it could result in them getting sick or dying or making it more difficult to survive. Sometimes it's completely silent, so it has no effect at all, and we don't even notice. We're going to look at a lot of harmful mutations today with the genetic diseases that we're watching in our videos. So, on top of that, mutations can also occur not only randomly when the DNA replicates, but because of chemicals. So we call these chemicals mutagens, And you guys may have probably should actually say probably have heard about. You would have heard a lot about UV light, right? Because you wear sunscreen. We're always concerned about skin cancer in this country because we don't we have an ozone hole. And so we wear the sunscreen to protect us from the UV light, so the UV light doesn't cause our cells to mutate and then give us skin cancer. We were talking about Rosalind Franklin in our last lesson and how she did a lot of experiments with X-rays and how then she got, I think, pancreatic cancer or ovarian cancer. And that was because of her exposure to X-rays. So that caused, again, lots of mutations in her body. Videos, and then we'll get to do some arts and crafts. So, there are multiple ways mutations can occur in different types. The first type is a point mutation, and that's just when one of the nucleotides change. So it just means I swap out one nucleotide for another. Now, depending on it, we can have three different possibilities. The first possibility is that it's a silent mutation. So even though the nucleotide has changed, it still codes for the same amino acid, and so nothing has actually changed in the protein. The second example is a missense, and in that case, my amino acid changes in that sequence. Sometimes this is bad, sometimes this is tolerable. It really kind of depends on what has swapped out. The last one, which is really problematic, is known as a nonsense mutation, and it changes the nucleotide to tell it to end the amino acid chain early. So instead of having a nice long amino acid chain, it ends prematurely and it's really, really short. And so it's basically useless. So that is point mutations. And we can see some examples in this image on the side where we see how it's been swapped out. In this case, the top one silent, it's still the same enzyme, still the same amino acid. The nonsense, the dashes means that it stopped making the amino acid chain. The missense, we just have a swapping out of a different amino acid. So that's the first type. The second type is a deletion mutation. And what happens is that one nucleotide actually gets removed. Now, this is a big problem because it's going to change the reading frame. So all of the amino acids afterwards are going to be wrong. The other one is an insertion, and that's when we add an extra nucleotide in. And again, both of these, very, very bad because it causes what we refer to as a frame shift. So the reading frame changes, and it means that everything afterwards, because we have to read it in a 3-set number, is going to be wrong. So I'll give you guys some examples using like an English sentence. So, the original sentence is time to dream. If I have a point mutation, I swap out the letter. So I swap the I to an A, and we have time to dream. Now that doesn't really make sense, but it still works. Yeah? Mutations, I could reverse the order. That's another one that happens sometimes. And again, it's just change the word. It makes a little bit of weird sound. But deletion and insertion, you see how it changed the full reading frame of the sentence, and now none of the words make sense afterwards. So that's the different types of mutations we can have, and they can result in different kind of magnitudes of mistakes. Cool. I think that's it for my notes, because now I think I have the videos to show you. Yes, I do. All right. So what I'm going to do is I'm going to show you 3 videos. And with those 3 videos, we're going to take some time afterwards so you guys have a chance to research what type of mutation it is, and also, you know, a little bit of information of why it's problematic. So I apologize. It's probably going to be a little bit depressing. These are very actually sad genetic diseases. So I'll plug in my sound. I'll pause the recording. So if people want to watch the videos, they can watch them separately on the Google site. So I wanted to show you guys these videos because I think it's a really good way to see how these genetic mutations really impact our lives in quite major ways. what was I going to say? A lot of you guys have mentioned interest in potentially going something in the healthcare medical field, and so I figured hearing about these things is really interesting. So before I play the next video, I want you guys to take a moment, hop on your device, and I want you to figure out what type of mutation causes sickle cell anemia. Now, there is space in your booklets for you to write these answers down, just FYI. So I want you to identify the type of mutation it is. So in the previous note, you can see it right here in the corner. There's a whole list of different types it could be. So I want you to figure out what type of mutation causes that sickle cell anemia. I also want you to think about what protein is affected. We were talking about proteins the other day. And what is that protein's role in the body? And we're going to ask the same question over and over again with our different types of diseases. So take a moment to figure that out, and then I'll show you guys the next kind of depressing video. All right, so what type of mutation was sickle cell? Anyone grab it? When they Googled it out of that list? It's just so quiet today. Just tired? Just everybody. You're so quiet, yeah. Sickle cell anemia is an example of a point mutation. So there was a change in the DNA sequence. It results in a change in the amino acid. And that amino acid change is really, really dramatic. In that video, you guys saw that the red blood cells should be round and oval shape. But in the case of sickle cell, that mutation changes the shape of that hemoglobin protein. And now the red blood cells are sickle shaped. They're kind of like a crescent moon instead of being an oval. I'll draw that on the board so you guys can see what I'm talking about. So what's the problem with it being a wrong shape? First off, it won't go through our blood vessels as well. It gets trapped and stuck, and then that can cause organs to die because it's cutting off the blood supply. That's what caused her stroke, and that's what also causes like different liver damage and things like that. The other thing is that this sickle cell shaped red blood cell doesn't carry as much oxygen as well. So it's quite problematic to have these cells. So she was going through and getting a blood transfusion and getting it swapped out. So they were trying to take out her blood, which had these defective cells, and put in a donor's blood, which had the right shape. So yeah, quite a sad thing there. But that's one example. The second example that I want to show you is cystic fibrosis. Yeah, these videos are going to be a little bit depressing because it's quite sad when you hear about these genetic conditions. So with cystic fibrosis, once again, just like with sickle cell anemia, I want you to take a chance to research the type of mutation that causes it. Like I said, we talked at the beginning of the point mutation, the deletion mutation, the insertion mutation. So I want you to see what mutation causes it. And I also want you guys to have a look again at that protein and think about what is that protein responsible for that's causing these issues. So we can see in the video, they talked a lot about the lungs being particularly affected. So have a chance just to kind of look that up, and we'll discuss it in a few minutes. All right. So cystic fibrosis is an example of a deletion mutation. In that case, I can't remember. It's like the cystic fibrosis transport protein or something like that. It's a really important protein in our body to help regulate salt content and the transfer of liquid. So if you don't have that protein working properly, what's going to happen is two things. The main one in the lungs that we think about is it's not able to regulate the water inside the lungs. So you should have some moisture in the lungs. But obviously that's not getting regulated properly. And so there's an accumulation of mucus. And that's what she was talking about and needing that lung transplant. And that's what the vest she had put on to help vibrate, to kind of get the mucus to break up so that way she could cough it out. Obviously, think about when you've been sick and you have like a chesty cough and that mucus builds up. It's really uncomfortable. It can be difficult to breathe. And that's what their life is like all the time because they don't have that ability to regulate the salt and water levels inside their lungs. I can't quite remember. I think it was I can quickly Google it. Let me check it. Let's see. There we go. Here we go. I'll just pull up the screen so you guys can see it. Let me just move it. Here we go. So here's cystic fibrosis. Cells that produce mucus, sweat, and digestive system. I might look up the mutation because it tells it in more detail. So I don't need it to be in like a lot of detail. I'm just trying to identify what type of mutation it is. So in this case, it's an example of a deletion. I know I found that somewhere else. Recessive disorder we'll talk about later. But the mutation is in the CFTR gene, and that's what's creating that protein. I look at the protein. There you go. Cystic fibrosis transmembrane conductance regulator protein. You just say CFTR. Yeah, CFTR. That is an amino acid. Oh, amino acid. Yeah. Yeah. So it's in a deletion, and that deletion results in, I don't know if it's a frame shift or if it's a whole amino acid that's lost. I'm not too concerned about that. The main thing is I just want to show you guys examples of genetic diseases. But it then causes that protein to misfold. So if that protein is, like I said, misfolded, it's not going to work. And so in this case, this protein no longer works, and the people can't regulate the salt amount. And then salt ties in with water transfer. And we'll learn that when we're doing our osmosis internal. But yeah, really kind of sad one. It also affects the digestive system as well. It affects how people absorb nutrients. But the main issue is going to be the lungs. And that's why a lot of them talk about having lung transplants, because the defective protein exists in their lungs. So they transplant a set of lungs of someone that doesn't have cystic fibrosis, so their lungs work properly. But as you saw in that video, the stats aren't great for it. It was 50% die within the next 5 years, even with a lung transplant. Hopefully those numbers are better now, because that's quite an old video. But it's still quite sad. And when we think about cystic fibrosis, people with that can die very easily from like the common cold or a flu or things like that, because they're constantly having that mucus in the lungs. So they're at really great risk of infection, and they're already compromised. So if we tie it in and we think about what's going on in our current climate with the COVID, all these things that we're trying to do, we're trying to protect people like the one we saw in the video with that cystic fibrosis, because if they catch COVID, they're likely going to die from it. In fact, if they catch a cold or a flu, it could kill them. So that's why we kind of have to think about these things. All right, the last one I want to show you is, which is a TV show that I watched growing up. I don't know if you guys have seen much of Scrubs, but it does a good job summarizing Huntington's. All right, so once again, like the other diseases, find out the mutation that causes Huntington's and also the protein that is affected by this mutation and what its role is in the body. Have a chance just to kind of explore, go down that rabbit hole, and then we'll keep going. All right, so what's the type of mutation? You guys find it? Nonsense. Yeah. It's repeated, so that's an insertion. So we have a repeated mutation of insertion, so you're getting extra nucleotides that aren't supposed to be there. Yeah? Got it? So we'll talk about inheritance, because he was saying he had a 50-50 chance of getting it. We'll talk about that when we do Punnett squares. So with Huntington's and sickle cells, and all of these diseases will have Punnett squares attached to it that we can explain what's the likeliness of getting those diseases. Does that make sense? So there's a difference between the inheritance factor and what causes the disease. Okay? So with Huntington's disease, I don't know it in a great detail. You guys don't need to obviously know it in a great detail, but the main kind of thing to know about it is that it's a protein in our brain and it misfolds. And what happens with Huntington's is that it's the accumulation of that misfolded protein that causes a problem. So you don't see the symptoms of Huntington's disease until later in life, until like your 40s, 50s, 60s. It's really hard to predict when the Huntington's disease will cause a problem. Does that make sense? So, and it's also really sad and dangerous because if it's occurring so late in life, you probably have already had children and then you risk passing it down to them. All right, so let's do a consider the following. We're running short on time. I want to make sure you guys have time for the DNA bracelet. With these genetic diseases, do you think there's a cure? Yes or no, and why or why not? Can we cure sickle cell anemia? Can we cure Huntington's? Can we cure cystic fibrosis? No? Why do you say no, Phoenix? Yeah, we're talking about our DNA, aren't we? And that's our code of life. And not only that, we need to change every single cell in our body to fix it. Or how many cells do we have? Or you can probably change the cells that are affected by it, so maybe the lung cells, but it is a really hard thing to change. It's not hopeless, though. People are trying to change it. So if you end up, I don't know if we do it in high school, but definitely at university, there was this new thing with gene therapy that they're looking at with CRISPR to try to slice in and fix the genes, or I should say fix the allele, because it's a specific allele that's the problem. Does that make sense? It's a whole new thing, because when I was at university, CRISPR wasn't a thing, and now it's being taught as part of your curriculum. But yeah, so it's quite a sad kind of thing to think about. All right, so let's wrap up with a DNA bracelet activity. I know I have a lot of bracelet activities. I think they work for biology. So let me explain how we do this. That's a really good way to reinforce that importance of that sequence. So let me just switch to my. All right. I'm going to get a piece of paper too. Find my markers. There we go. Okay. So, ready? Okay. So, we spent quite a bit of time talking about the importance of the DNA sequence and how a change in that DNA sequence can change the amino acid, which can have downstream effects to then change our protein and thus change the phenotype. To reinforce that kind of idea of that linkage, what I have here is an alphabet, and that alphabet has each letter assigned a codon. So what we're going to do is take our names and translate them into a 3-digit code using that DNA sort of stuff. So I'll do a little bit of an example to show you what I mean, and then you guys will have the rest of the time to work on this. I'm happy if you just want to make your first name. You can do your first name and last name. You can do a nickname. It doesn't really bother me. We will be making a loom band bracelet based off of it, which would be really neat because then you can wear it and then it's actually your name in the code, which I thought was kind of cool. So I'll just do my Carrie, which is my first name, just because it's short to show you as an example. So I look for the K, and then I see with K, it translates into A, G. Then I look for the E, and the E translates to G, A, G. Then I look for my R. The R translates to C, G, T. And then I look for the I, and that translates to A, T, and A. So now I've taken my name and I've given myself a DNA sequence for it. Yeah? All right. So let's talk about how this then ties into the loom band. With the loom bands, I have assigned color in order to make our little bracelet. So let me go grab my loom band. So for these loom bands, any A is going to be a T. Any A is going to be red. Any T is going to be green. Any G's are going to be purple. And then for the C's, because I don't have quite enough of both either color, you can choose to do either pink or blue. Pick one of the two. So pink or blue, I'm not fussed because I don't have enough of one or the other. So get the DNA sequence and then grab the colors that you need and then start building that loom band. Mindful of the time. So I suggest when we're doing the loom band that you grab 2 pens because it makes it easier to put it on. So I have A, and D. So I'm going to need red and I'm going to need purple. And what I'm going to do is I'm going to put the colors in that order for my bracelet. So the first one that we put on, we need to twist. We're just going to do a simple loom band. Put that on, twist it, put on the second one. There we go. You can also do this with your fingers, but I figure it's easier with the pencils or pens. So the first one I grabbed was red, and then the second one I have is red as well. You start by putting 3 on. The next one's a G, so that one's going to be purple. So I put a purple one on next. I hope so. If not, we can always do it our next lesson. To get started, we can at least get our names translated so that way in our next lesson you can build it. And then we pull upward, and that's what starts it. Then I keep looking. I have a G, so that's going to be a purple. I put a purple down, and then I pull it up like that. Does that make sense? And then back to the A, so I put a red one, G, then I'll put a purple one, and then we keep going until we have a chain. And I have some filler ones in case it's not long enough to make a bracelet as well. But I think we only have 10 minutes left. How about we translate the names into the codes, and then that way when I see you guys next week, we can dish out the colors, and you guys can make yourself a little loom band. Does that sound like a good plan? All right. I'll test these guys out. I would suggest find a page in your notebook so that way you have it ready for next week. All right. Hey. Listening up, put away the phone. I would suggest, because otherwise you're just going to have filler. What else did I want to say? Go ahead. All right, so we have about 8 minutes left. So make sure you have your code ready because in our next lesson we'll actually assemble the loom bands. But to summarize today's lesson, we were defining gene and allele, and we should also be able to explore how mutations create those new alleles. Off the success criteria, you should know the difference between gene and allele. You should be able to identify that mutations are the source of the gene pool. Oh, don't put up the chairs yet. You should be able to define mutation and know the different types of mutations. You should be able to explain how mutations affect an organism. Obviously, we saw a lot of genetic diseases, and you should be able to, oh, we didn't talk about that one. We'll probably do that one in another lesson. My mistake. We shouldn't do number 6. All right. If you want to leave your books, feel free to.